want to start by acknowledging a number of organizations that we've worked with over the years, in particular the BC Shellfish Growers Association, Taylor Shellfish Farms, the Hakai Institute, and Vancouver Island University. Okay, it doesn't seem to be, oh, there we go. Okay, just to introduce the subject of climate change and the cause, I'm sure everybody knows this, but just so that we're all on the same page, we have UV rays coming from the sun, causes the earth to warm up. Some of those rays are reflected out through the atmosphere, but when you get more greenhouse gases accumulating, those greenhouse gases block those reflected rays back down to the Earth's surface and thus warm the Earth's surface. So that's kind of what's going on generally in the atmosphere. More specifically, what's going on in the ocean. Well, the oceans absorb about 90% or more of the heat that's generated in the atmosphere, so they're definitely warming up. With that, there's a concomitant decrease in oxygen because the warmer the water gets, the less oxygen it can hold. And along with that, because the oceans are also picking up atmospheric CO2, and they take up about 25% of the atmospheric CO2, the oceans are becoming more acidic. So there's three variables here that are all changing quite rapidly, and they're having major impacts on the oceans. And you can see the six boxes at the bottom of the slide there. We're seeing sea level rise, which is accelerating flooding in coastal communities. We're seeing bleaching of warm water corals in many parts of the world. We're seeing increased toxic algal blooms in various areas. Lower oxygen levels are suffocating some marine animals in their habitats. More acidic water is harming organisms that have shells, and then disruptions in fisheries and marine food webs as well. So there's a lot going on with climate change in the ocean. Now, if we look specifically at ocean acidification, the left panel here is basically what's going on when waters are less acidic. The right panel here is when waters are more acidic. So when the waters become more acidic, more carbon dioxide is going into the waters, you get this focus on producing carbonic acid, free hydrogen ions, which drives down the pH, and bicarbonate ions, and less carbonate ions, which are used by your marine calcifiers. So corals, shellfish, all kinds of mollusks. So more CO2 in the oceans are driving things more this way and less this way. And when we're talking about shellfish, we usually talk about two forms of calcium carbonate, aragonite and calcite. Aragonite is used by the younger shellfish, typically the larval shellfish and early juveniles, and then they switch over to calcite as they get older. And we often talk about a saturation state of aragonite or calcite, which is really kind of the concentration. I won't get into the details, but when aragonite calcite saturation is less than 1, it's a very big problem for shellfish forming organisms. They have difficulty in forming shells when the saturation states are that low. So when we talk about climate change, and specifically in the ocean, we think about a number of things. We think about warming waters, more acidic waters, and lower oxygenated waters. And the three graphs you see here are the RCP or SSP projections out to 2100. RCP is representative concentration pathway. SSP is shared socioeconomic pathway. These are model projections of what's going to happen to these variables in the long term, in this case, out to 2100. And there's different models that show different kinds of projections depending on what we do as a species, humans. So you can see in this middle graph of pH, SSP1, 1.9, 2.6, 4.5, 7.0, 8.5. Generally, the lower numbers are best case scenario. So, RCP 2.6, for instance, which is one of the better or best case scenarios, assumes a future with strong mitigation efforts by humans, so that we have a peak in relative forcing around mid-century and then a decline to 2.6 watts per meter squared by 2100. Then RCP 8.5 assumes kind of the worst, that emissions continue to rise. We can't get the emissions down at all, leading to a radiative forcing of 8.5 watts per meter squared by 2100. And you can see by looking at these various graphs what's going to happen. With temperature, this top graph, we have a projected increase of 0.3 to 1.7 degrees Celsius under the best case scenario, and 2.6 to 4.8 under the worst case scenario. That's by 2100. pH, we show a decline of approximately 0.12 units by 2100 under the best case scenario, and about 0.5 units under the worst case scenario. And you can see other typical trends here with DO as well, declines based on the RCP scenario that you're using. So that's what's projected to happen long term in these three variables in oceanic waters. But of course, we have shorter term stressor events that are occurring on top of these long-term patterns. So these are some data from Wiley Evans at a Hakai Institute paper that he wrote back in 2019. If you look at these two graphs here, the top graph is aragonite saturation state, and the bottom graph is pH. And the x-axis shows over time. And basically what he had shown was that you get these low pH events occurring during the winter months. You can see these bottoming outs here and here and here. And it can get quite low pH, so much so that at certain times you're looking at aragonite saturations which are less than 1. Which is problematic for shell-forming organisms. But he's also shown that you can get these high CO2 events during summer months as well. And this shows depth on the y-axis versus aragonite concentration on the x-axis. And you can observe aragonite saturation states under 1 quite near the surface during summer months as well. And these upwelling events of low pH water, we have lower pH water at deeper depths, and sometimes it gets upwelled, causing these acidic upwelling events. These upwelling events of low pH acidic water have been connected to high larval mortality in shellfish hatcheries over the years. This has been a common occurrence, and hatcheries have actually started using buffering their seawater in order to bring up the pH of their hatchery water in order to get proper larval development. So this just shows other kind of short-term stressor events that we have in the world's oceans. The left-hand graph here is the heat dome that we had back in June 2021 here on the West Coast, where temperatures got very high. The right-hand graph shows that you can have O2 declines to less than 2 milligrams per liter in various sites around the world's oceans. So you have both heat wave and hypoxic events occurring in different areas of the globe. Now to switch gears a little bit on to what we work on in our lab. We primarily work on the Pacific oyster. Scientific name is Chrysostrea gigas. We have started getting into newer research looking at climate change impacts on pteropods and razor clams, but I won't be discussing any of that research today just because there's so much to talk about with Pacific oysters. And the reason we chose this particular species is it supports regional and indigenous economies. There's both a wild fisheries and a quite valuable aquaculture industry with the species. Of course, it is used for food, social, ceremonial purposes for various First Nations. They provide ecosystem services. Wild oysters occur oftentimes in reefs or beds that are habitat for a variety of marine invertebrates and fish species. They are filter feeders, so they filter the water column and take out particulates and debris. Um, and the other reason we're interested in looking at them is that they're particularly vulnerable to climate change impacts because, first of all, as juveniles or adults, they're non-motile. So they can't get out of an area when there's a heat wave or a low pH upwelling event. They have to stay put and deal with it. They're an intertidal species, so they're out of the water a couple of times a day for various periods of time. During warmer weather, they can be baking in the sun during when the tide is out. They're thermoconforming, so they basically take on whatever temperature of the water. Of course, they have calcareous shells, so when we're talking about ocean acidification and aragonite or calcite saturation states less than 1, and they have problems making their shells. And generally, what we look at is multi-stressor impacts. So we're interested in learning what are the impacts of both warming and ocean acidification on this species, rather than looking at individual effects of either warm water or acidification. So, and usually what we measure are things like growth, survival, respiration, and a big part of what we do is looking at the gene function. What genes are getting upregulated or downregulated in response to these stressor events. Where we work in the field, we work at various places, Discovery Islands, Baines Sound, Barkley Sound. We're also working with the Haida Nation up in Haida Gwaii. That's the razor clam stuff. But mostly we're located in Baines Sound when we're doing our field work. In the laboratory, we work at the Pacific Biological Station in a lab, which we call FOCAL, FOCCOAL, the Fisheries and Oceans Climate Change and Ocean Acidification Laboratory, pictured here on the left. And it's a system that's it's automatic. It's controlled by a programmable logic controller, which talks to mass flow control valves, which control the amount of heated and cold seawater and CO2 and air mixing so that we can produce waters of various temperatures and various levels of CO2. And it's all controlled automatically. And it's a flow-through system. It's not static. And so usually we do our experiments in containers like these 23-liter buckets here. These black things are insert trays that you see here in this photo. And you can see the oysters in that. And then each one of these experimental units has its own header tank that flows directly to it. Um, and we produce the temperature and CO2 concentration that we're looking for in these header tanks. When we talk about, looking at a species' vulnerability versus its resilience, you can think about three things. You can talk about the exposure, what is the level of the stressor and how long is the organism exposed to that stressor. You can also talk about adaptive capacity. So can that species evolve over time to deal with that stressor? And then there's also sensitivity. How sensitive is the organism to that stressor? And can you do something to make the organism less sensitive to that particular stressor? So for exposure, you can alter or alleviate the exposure conditions and reduce the coinciding stressor impacts. To deal with sensitivity, you can improve the species' ability to respond and recover from stressor periods. And for adaptive capacity, usually we're talking about selective breeding to make individuals more less impacted by stressor conditions. So you're manipulating their adaptive capacity. So the rest of my talk is sort of going to be broken down into these three circles here, and I'll talk a little bit about what we've been doing in our lab to address these three issues. So I'll start with adaptive capacity. This is work that we've been doing in conjunction with Dr. Tim Green at Vancouver Island University, and he's really the one that's spearheading this. We originally started, I want to say probably 8 years ago, maybe more, with a DFO funded project looking at producing various families of Pacific oysters and seeing if they dealt with things like warming water, low pH, differently. So were some families more adept at dealing with stressor conditions than others? So a lot of this work has been done up at the Deep Bay Marine Field Station of Vancouver Island University, which is of course in Deep Bay. And Tim does all of his work using pair mated families. And he's now, I think, probably at, I want to say F4 or F5 now, 5 different generations of families. This was early work that we did where we had 25 different families. This is showing percent of larvae on the y-axis versus the different families on the x-axis. This graph here is under ambient conditions, and this graph here is under aragonite undersaturated conditions. And the different bar colors are different types of larvae. So a normal shaped larvae is the blue color. Those that are not normally shaped or abnormally shaped are the cupped ones in red, the convex hinge ones in yellow, indented margin ones in green, protruding mantle in red or orange, sorry, trophophore and gray, and egg in black. So those individuals that aren't even getting to the larval stage, the eggs and the trophophores. And what to notice here is on this graph, which is under ambient conditions, You have families that are doing better or worse. There's definitely differences there amongst the different families. And you also see that under aragonite undersaturated conditions. You see some families doing fairly well. This family here, right here, showing a large blue bar, normal shaped larvae, versus something like this, not doing so well, or these three families here are not even getting to a larval stage. So there's definitely different tolerances to pH conditions based on family. These were the same family of oysters, but now we're looking at juveniles over time. I was during a 1 year grow out at Deep Bay. So this is percent survival on the y-axis and the different families on the x-axis. So we see that survival after 1 year grow out is dependent on family, as low as about 40% up to 100% survival after 1 year. Again, this is looking at the same families, and we're looking at juvenile oyster survival on the y-axis. But it's after a 120-hour challenge with a bacterium called Vibrio asterianus, which is a bacterium that has been shown to be involved in summer mortality incidences of Pacific oysters. So it's a pathogenic Vibrio for Pacific oysters. And here we can see that different families of Pacific oysters deal differently when exposed to Vibrio. You can see survival as low as about, 5% up to 70% after a 120-hour exposure with this species of bacterium. This is another experiment with the same families of juvenile oysters. Just focus on the black bars, which are percent survival on the y-axis, and the different families on the x-axis. And this is another challenge with Vibrio asterianus. It's a 96-hour challenge. And again, you see different survival rates depending on the family. As low as about 8%, upwards of greater than 80% based on the family. I think one more slide here on the selective breeding aspect. This is different families. I think this is an F3 or F4 generation now. So this is further along in Tim's breeding program. This is showing 19 different families under ambient conditions, which are the blue bars, and aragonite under saturated conditions, which are the yellow bars. And different families are on the y-axis here. And on the x-axis is absolute growth rate per day. So grams per day, that's wet grams. And you can see that there's different families that perform better under both ambient and aragonite undersaturated conditions. And there's a wide range of percent survivals, or sorry, absolute growth rate dependent on families. So that's some of the work we've been doing on selective breeding, and Tim Green at Vancouver Island University continues that work, and continuing to produce families that are more tolerant to low pH conditions or warming conditions or exposure to pathogenic bacteria. So that is work that's ongoing. If we move on now to talk about the sensitivity of the species, how can we change an organism's sensitivity to warming water or ocean acidification? So this was a rather large experiment that we conducted a few years ago, and it was basically it originated based on what growers had been reporting to us in a number of meetings, and they were saying that typically they would find that their intertidal populations were doing better than their subtidal oyster populations when there was a heat wave. Um, and that was kind of you might think it's backwards. You think that intertidal populations might be more stressed out during warmer periods, but we got to thinking perhaps the intertidal oysters build up their stress tolerance over time because they are left without water for certain periods of time daily when the tide goes out, and maybe they build up a stress tolerance based on that. So we designed this experiment where we had two populations of oysters at three farm sites. Um, One population was subtidal for both year one and year two. The other population was intertidal in year one and then moved to subtidal in year two. And in years one and two, we tracked survival, growth, and condition index at the three farms. In year one, we had a laboratory challenge where we compared survival during acute warming. In year two, we had a laboratory challenge where we compared survival during acute warming and Vibrio challenge. And then in year two, we compared microbiome and gene expression response in relation to a natural heat wave event. This is just a graph showing temperature data at one of the farm sites, showing that there's vast differences in what the temperature is doing at the subtidal and intertidal locations. In year one, or at the end of year one, we found no significant differences in survival between the intertidal and subtidal treatments at any of the three farms, and there was no significant differences in shell length or condition index between the two populations as well. That was at the end of year one. Um, at the end of year one, we took individuals of both populations and we exposed them to a stressor experiment in the lab. So we ran an experiment where we subjected oysters to 16, 20, and 24 degrees Celsius for 10 days under constant immersion or constant immersion. Immersion, IM, is always in the water. EM, immersion, is out of water. Um, and if you look at this graph here showing survival probability on the y-axis versus time on the x-axis, you'll see that at 20 degrees Celsius, intertidal oysters had significantly greater survival than subtidal counterparts, so 100 versus 90% at day 10. And under immersion, or EM, intertidal oysters had greater time at 100% survival than subtidal counterparts, 4 versus 2 days. And I just showed one temperature here, 20 degrees Celsius, but results were generally similar for 16 and 24 degrees Celsius as well. Um, this is the results of after year two on survival, growth, and condition index. This graph here is showing percent survival, and only one farm site. We had higher survival, approximately 20% increase in intertidal than subtidal oysters. Remember, when I say intertidal, I mean oysters that were intertidal in year one that had been transferred to subtidal in year two. So those oysters had 20% increase in survival, but there was no significant difference in shell length or condition index between these two treatments. At the end of year two, we did another laboratory stress experiment where we subjected the oysters to heat stress and Vibrio, Vibrio asterianus. So we ran the experiment at two temperatures, 16 and 24 degrees Celsius, with and without the Vibrio for 13 days. This graph shows the survival probability over time for deep and intertidal control individuals and deep and intertidal Vibrio exposed individuals. At 24 degrees, we had no significant differences in survival between intertidal and subtidal oysters, either with or without Vibrio, because most of them died at that temperature. But at 16 degrees Celsius, which is this graph here, we see that when they're exposed to Vibrio, the intertidal oysters had increased survival time and greater overall survival after 13 days than the subtidal oysters. This was another experiment we did in year 2, looking at oysters that were exposed to a heat wave, so short-term stressor event. We looked at gene expression and microbiome of the oysters. And the question really was, does culture environment, intertidal versus subtidal, alter microbiome and gene expression in relation to a heat wave event? So we compared gene expression and microbiome of both treatments, intertidal and subtidal, at one farm. We looked at the microbiome at 3 time points pre-transfer, pre-heat wave, and post-heat wave, and we compared the oyster gene expression at 3 time points pre-heat wave, heat wave, and post-heat wave. If we look at the gene expression results, there was a significant effect of time on gene expression, so higher expression post-heat wave, but no significant difference between the intertidal and subtidal treatments. However, if you focused just on the post-heat wave time point, there was a significant effect of culture treatment on gene expression. And we saw significant differences in the expression of a few genes, including superoxide dismutase and alternative oxidase. What was going on with the microbiome during that heat wave? The microbiome richness was significantly higher in gills of the intertidal oysters than subtidal oysters. And the microbiome richness significantly changed over time. You can see in this graph here where we're looking at microbiome richness in the pre-transfer, the pre-heat wave, and the post-heat wave samples for individuals that were in the intertidal, transferred to the subtidal year 2, and individuals that were just in the subtidal for both years. And you can see the change in microbiome richness between intertidal and subtidal in pre-heat wave and post-heat wave samples. This is some work that we're currently doing. I don't have any results yet, but this is a trial where we're looking at combining short-term stressor events on top of longer-term climate change impacts. So we have oyster populations that were primed under various conditions. So one population was completely immersed in seawater 100% of the time. Another treatment where oysters were taken out for 4 hours per day, every day over a 2-month period. Another treatment where oysters were taken out of water for 24 hours every week for a 2-month period. Those 3 populations of oysters are being subjected to a long-term climate change. So 20 degrees Celsius predicted future warming versus present day temperature of 16 degrees Celsius. And then they are going to be subjective to heat waves or hypoxia events for a 2-week period during that long-term climate change period. We have oysters that are both in the lab that are subjected to these man-made extreme events, and then we have oysters that are outplanted to the PBS farm site. that are out in the wild, which we will be sampling as well. Also, that finishes the work that we've been doing on sort of changing the sensitivity of oysters. And now I want to finish up with exposure. How can we reduce the exposure of oysters to extreme conditions? And this was work that was sort of born out of what they're doing on salmon farms when they have low oxygen conditions or conditions that have um, high levels of toxic algae. They bring out these bubblers, artificial aerators. And you can see on this slide the aerators to the side of the cage, and it's kind of acting as an artificial barrier of this toxic algal bloom. But you can also have bubblers that are underneath finfish cages that presumably bubble up maybe colder waters that will cause less stress for the finfish as waters heat up. Um, and we're thinking about doing this with oysters as well. And this isn't anything novel. This was done by some Japanese researchers back in 2014. This was a field experiment that they had where they had artificial aerators under trays of oysters. And they did find that aeration can improve bivalve culture if it is performed at a rate that overcomes hypoxia formation and high water temperatures. Um, so you have to have the aeration set at a certain level that it's going to overcome that stressor. So this was a short-term experiment that we did in Focal. This was some work of a graduate student of mine, Andrew Bickle. This was very short-term, over 10 days, and he looked at a number of genes. HSP90 was one of them, but he looked at, I think, about 12 or 14 different genes of interest. He had basically three different treatments. One was a control treatment of 16 degrees Celsius. Another was a heat wave that went from 16 to 26 degrees Celsius over the 10 days. And the third was heat wave and hypoxia. So the red bars were a treatment where it got hotter, but there was aeration. And the purple bars, it got hotter, but there was no aeration. Okay, and you can see the levels of dissolved oxygen over here in the various treatments. And basically, what I want to highlight here is at day 10, we see this significant increase in HSP90 expression in the heat wave and hypoxia treatment, showing that this particular gene is getting upregulated when there's a hypoxia and heat wave co-occurrence. Um, so this has got us to thinking, and this is some work from another graduate student of mine, Sierra Gray. I just put this figure here to sort of talk about what we envision might happen in the future in terms of using these genes of interest to help alleviate stressor conditions on oysters. We've looked at a number of genes of interest now that get turned on or turned off in response to warmer waters or higher CO2 concentrations. And we're hoping that at some point we will be able to have a gene of interest that is sort of an early warning trigger system. And this will take a long time because these sort of analyses take a long time to do and they're expensive. So it's not something that will happen in the near future, but hopefully someday we'll be able to look at oysters on a daily basis and look at what kind of genes are getting turned on or turned off and knowing before oysters get too stressed out that, hey, we need to do something about this. And maybe we need to, turn on artificial aeration or submerge the oysters deeper into colder water. And that's sort of the thinking behind this. One more thing I want to talk about in terms of alleviating exposure conditions. This was some early work we did on larval rearing of oysters in soda ash buffered versus ambient pH water. And at this time, a lot of hatcheries were using soda ash to buffer their incoming seawater to reduce impacts of low pH water on their larvae, and it seemed to be working. Um, but we looked at survival of juvenile oysters and adult oysters that resulted from larvae that were either produced under buffered soda ash buffered conditions or under ambient conditions. So those that weren't buffered. Um, and they're either exposed to Vibrio asterianus, pathogenic bacteria, or they weren't a control. And basically, what you can see here is those individuals that resulted from larvae that had been buffered in the hatchery did much more poorly than their counterparts that hadn't received the buffering treatment in the hatchery. This is just showing survival probability over time and days. This is the survival curves with the juvenile oysters, and we showed similar things with the adult oysters that resulted from those larvae as well. In that those that resulted from larvae that were buffered in soda ash treated water did more poorly than their counterparts that weren't under buffered conditions when they were later exposed to Vibrio asterianus. So there's something going on there that, when you buffer the seawater in the hatcheries, you may be affecting later survival of juveniles and adults in the field. And this just shows the different microbial groups in the larvae in the hatchery that were exposed to ambient conditions and those that were exposed to the soda ash buffered conditions, and then the juveniles under ambient conditions and the juveniles under buffered conditions. And you can read here that there's differences in the number and types of groups and the percentage contribution of those different groups based on whether they were under ambient or buffered conditions. So to sum up here, when we talk about manipulative adaptive capacity, we've been doing this with Dr. Tim Green at Vancouver Island University over a number of years, and he has a number of F generations now, which he has produced over the years. I think he's up to 4 F5, and we do see that there are some families that do better under ocean acidification conditions, and some families that do better when they're exposed to pathogenic bacteria. Um, we've learned that you can decrease sensitivity to field stressor conditions when you grow your oysters in the intertidal prior to putting them out in deep water, but it's not consistent among sites. So we only showed this at one of 3 sites. So we'd like to do some more research there, and that's kind of the basis of the uh, laboratory experiment that I showed you that we're currently running that has the heat wave stressor overlapping the long-term climate change stressor. And then we're interested in following up more about using artificial aeration to reduce stress response during high temperatures or low dissolved oxygen conditions. So we have another experiment on that this fall. There's a host of people to thank here who we've been working with over the years. I won't mention all of them. They're all detailed here. I would highlight Clara McKenzie and Chen Walker that work with me currently at DFO, Tim Green and Sarah Leduc at Vancouver Island University. Really want to thank Alex Monroe at Taylor Shellfish Farms, Nico and Malcolm at the British Columbia Shellfish Growers Association, And all of our microbiome work is done through the Hakai Institute. So I'd like to thank Colleen and Root for being involved in that work. Most of the funding for this work over the years has come through DFO, either through the Aquaculture Collaborative Research and Development Program or the Competitive Science Research Funding Program. But we've received funds and in-kind support along the way from the BC SGA, Taylor Shellfish Farms, Vancouver Island University, and the Hakai Institute. I want to thank you for your time listening to me and open to taking any questions if I can answer them.
Awesome. Thank you so much, Chris. Appreciate it. I know there are a lot of great resources in the chat that I will share out, from books to documentaries, to some of these papers
Great.
as well. If folks want to drop questions into the chat, they can. Otherwise, if you want to raise your hand, we'll also call on you and you can ask live. Our first question from Peter is whether you expect some parallels with some of the native bivalves.
In terms of impacts?
I would say maybe in terms of impacts or in terms of strategies to mitigate those. Take your pick.
Yeah, so in terms of impacts, yes, I would expect the same sort of results. And we have a current project where we're looking at the health and genetic expression of Olympia oysters, which are a native species in BC and Washington State. So we're going out and sampling them for health status, and we're looking at genes of interest, hopefully looking at how those genes of interest get upregulated or downregulated in response to maybe a warming event. I'm expecting sort of similar type impacts on other shellfish, although there would be different impacts on, say, maybe subtidal organisms like geoducks that don't occur in the intertidal and live in the sediment. So maybe warming wouldn't be so much of a concern for them. In terms of mitigation, that's a good one. It's difficult to mitigate any of these stressors in terms of wild populations because you're not really containing or controlling them. So the only thing that I can think of is if you're doing some restorative work in terms of, boostering or bolstering wild populations with hatchery produced individuals, or bolstering wild populations for fisheries. So the geoduck fisheries folks do that. So, it's the same sort of thing with selective breeding. And can you build a better organism that is better able to withstand warming or ocean acidified conditions? But that's, I think, basically all you can do in terms of wild populations.
Great. Appreciate those reflections. We've got another question, which is about how this research moving forward might integrate traditional ecological knowledge regarding mariculture in terms of clam gardens, or help inform, and you touched on restoration just a moment ago, but new construction of cultural clam gardens in particular.
Yeah, that's a great question. In terms of clam gardens, uh, if you were stocking those with hatchery produced seed, again, you come back to the issue of, selective breeding for tolerance to stressor conditions like warming and ocean acidification. Um, I mean, we have been working quite a bit with First Nations in the last few years. The Hakai, or sorry, the Haida Gwaii First Nation is interested in, declines in razor clam populations in their territory. So, they're questioning whether that's linked to low recruitment because of climate change. So we're doing work with them. Um, yeah, I don't know if that answers the question or not.
I appreciate it. And if others in the chat too have thoughts
Yeah.
on connections to other projects or research related to that, please feel free to drop them in too. Um, that's the beauty of getting to collaborate and discuss. We also have a question just around mussels and if there's any researchers in the area or more broadly who you would point to who are thinking about similar sorts of impacts and research for mussels.
Um, hmm. Not within DFO in BC. Uh, can't think of anything. I know Chris Harley was on the line. I don't know if he still is. I don't want to put him on the spot. I don't know if he's doing anything. He might be able to answer in the chat if he's doing anything with mussels. We are not.
Okay,
Chris is at UBC and he does a lot of climate change work with marine invertebrates. So yeah, he might be able to answer your question. I know for sure nobody in DFO Pacific region is working with mussels in terms of climate change impacts.
totally fair. It looks like Chris had to drop off, but maybe I'll shoot him a note afterwards and
Okay,
just if he does have any recommendations, again, I'll include that in the follow-up email to folks.
sounds good.
Cool. Other questions? Again, feel free to raise hand too. While folks are thinking, I will just do a shout out. I dropped this in the chat, but for the summer, we'll take a pause and a breather for the roundtables. And so, if folks have recommendations for this fall of topics they'd like to hear more about, feel free to drop those in the chat or email me. The other thing that we did last year to add some serendipity to our virtual worlds and connections was coffee chat roulette. So you submit kind of where you're based, what you're interested in, and I'll do some matchmaking on the back end. And then from there, once you're paired with someone, you have the flexibility to connect when and however it is most convenient for you. So if anybody is excited about that, July 31st is your deadline to submit that form there. Okay, did I buy any? Ooh, there we go. Yep, bought some time. So two questions. First is
Mhm.
Vibrio vitality flat across the experimental temperature range?
Sorry, could you repeat that?
Yeah, is Vibrio viability, sorry, flat across the experimental temperature ranges?
Oh, that is a great question. Um, I am not sure. I'd have to think about that a little bit more and delve back into that research. I'm not convinced we looked at that issue when we were doing the experiment, but we might have. But that's an excellent point. Yeah.
Great question.
Uh, who asked that?
Yeah,
Can I know?
yeah, of course. David Mackus asked that.
Okay.
Yep.
A great question.
And then we have our next question, which is just whether there's potential for multi-trophic aquaculture to mitigate some of these impacts. So co-culture with algal species as an example of that.
Yeah, we ran a sort of a preliminary laboratory experiment where we subjected oysters to acidified conditions and uh, we either held them by themselves or held them with kelp blades or with sea cucumbers. There's some evidence that tropical sea cucumbers, through their feeding habits, can reduce local OA conditions. Um, but that experiment was not entirely successful. And so we kind of really don't trust the results from it. But theoretically, yes, macroalgae that are taking up CO2 might be able to reduce local ocean acidification impacts. That has been shown in some Chinese work, both in uh, laboratory systems and mesocosms, and I believe even at base scale levels. Uh, but they're operating on a totally different scale to what we operate here in Canada. I mean, they have bays that are full of oysters or kelp or whatever species they're working with. So yeah, theoretically it could be done in BC. It has been shown to work in China with different kinds of macroalgae and shellfish species. Yeah, that's a good point.
Great. We've got another question here from Scott. He says, perhaps I missed this, but given that the results at 24 degrees Celsius, are you rerunning at intermediate temperatures?
Um, we hadn't given any thought to that, but it's a good suggestion. Um, Something not quite as brutal. Yeah, so maybe 22 or 21 or something. Um, but the 24 is not kind of a shot in the dark. It's kind of based on our observations that we've done in Deep Bay over the years and what surface waters are getting to during heat wave events. Um, so that's why we chose 24 for that particular experiment. We know that oysters that are held in deep water near the surface are probably going to be exposed to that kind of temperature during heat wave events. Um, but yeah, it would be interesting to know what the results would be using sort of temperatures lower than that. But we don't have any plans for that work yet.
Thanks, Chris. Appreciate that. Hard to see how warm it's getting in some of these places.
Yeah, yeah. I mean, I'm quite concerned, uh, knowing the rate of change and all the variables that are changing at the same time. Um, it is very concerning. Yeah.
Share that concern as well. And appreciate the research that you were doing to look at the interactions of these stressors so that as we're thinking about impacts, we have our eyes wide open, but also a good sense of how they interact. So we're not just focusing on one piece and saying, woohoo, we solved for that. And a year later going, oh, shoot. So particularly the combination of the temp and the bacteria is really fascinating. Well, I know,
I would put out that if I had, yeah, I have
go for it.
maybe 30 seconds here. Yeah, when we're running these experiments, like the one I said we were running right now, we're overlaying heat waves or hypoxic events on top of long-term stressor warming. They're really difficult and time-consuming to run, and we can't do everything. So, if people are looking to do some sort of measurements on organisms that are exposed to these kind of conditions that we're not doing, feel free to reach out to me. So typically, like I said, we do stuff on growth, survival, respiration, uh, gene regulation. But if folks are interested in getting samples for some sort of tissue analysis, I don't know, fatty acid, protein, carbohydrate, or shell strength, certainly reach out to me because, yeah, we could provide you with some samples perhaps that you could analyze, and that would give more meaning to the experiment that we're running. So yeah, I'll put that out.
Yeah, love it. Uh, those sorts of opportunities to take access to some of the samples as well as access to the analysis can lead to some really interesting insights. So welcome that. Well, I appreciate the really robust and thoughtful questions today.